America. It's a real pleasure to be here. And thank you all for making it today on a lovely morning in Florida. I joined Bruker in 2017. Before that, I did my postdoc, not quite close here, but at Scripps, Florida. Now I think it's UF Scripps, Florida. Is that what it is? So I think we're all related in certain ways. Today, I'm going to talk a little bit about our flagship product, the timsTOF line of systems, as well as trap time mobility technology, and how it makes a big difference in proteomics, metabolomics, lipidomics. And my colleague Kate Stumpo is going to tie it all around with MALDI-based imaging and how all of that comes together in what we call SpatialOMx. As Griff pointed out, please put your questions in the chat, and then we'll all address it together at the end of the day. Good. So let's get started. So Bruker launched its first timsTOF not too long ago in 2016. It's the timsTOF Classic. And from then on, the real timsTOF Pro was launched in on 2017. Pro initially because it was directed towards proteomics, but as you will learn today, it is basically a Swiss Army knife of mass spectrometers that can do proteomics, lipidomics, metabolomics, and MALDI-based imaging. So in 2017, we had a lot of success with the timsTOF Pro. That's one of the reasons I'm here, and it's one of the reasons you have the system and it's doing so well. So much so that in 2019, we launched the timsTOF Flex. The Flex is great because it's got a dual source. It's got a MALDI source on one end. It's got an ESR source on the other. You can do imaging, high resolution imaging, and then take that sample and run it on the system for metabolomics, proteomics, and lipidomics. A true multi-omics system. As what you guys pointed out, you do a lot of single-cell genomics, and this system really plays into the proteogenomics space. In 2020, we launched the timsTOF Flex with MALDI 2. It's a secondary laser that Kate's going to talk about later on. And finally, in 2021, we launched the timsTOF Pro 2. We're not happy with the Pro. We went one step further, launched the timsTOF Pro 2 with a better TIMS cartridge and better ion optics for better dynamic range and sensitivity. We also launched the timsTOF's SCP. The SCP stands for single cell proteomics, but it's ultimately a ultra high sensitivity system, which for any workflow where you're sample limited, that is the system to go. And I'm going to talk a little bit about that later on this talk. And finally, in 2022 ASMS, we launched the timsTOF HT. HT stands for high throughput. It's got higher dynamic range. You can do a lot of high throughput biofluid workflows such as plasma proteomics, serum proteomics, and CSF proteomics. So it's got a lot of potential, obviously, on the metabolomics and lipidomics fields too, that my colleagues will talk about. And finally, we also launched the timsTOF Flex MALDI 2 with microgrid that Kate's going to talk about. So this is the evolution of the product line, coming from the Pro initially over 5 years, all the way up to the HT, and we're just getting started. If development is the S curve, so we're just on the rise right now. We have a lot of innovations coming up, but my talk is primarily going to be based on what we do on the Pro 2, and obviously the HT and the SCP. And we've had a lot of success over the years with proteomics in general, and this is the gallery of the greats, as I call it. You can see people been doing proteomics for a while. Dr. Rudy Abersold at ETH, Dr. Nikolai Slapov with single-cell proteomics, and we have a good mixture of academics from universities, as you can see, as well as people from pharma. We have the director of proteomics for Bristol Myers Squibb, Dr. Chuck Dongre, as well as director of proteomics for Janssen, Paris Beltamin, actually, who is a Florida native that we know right now. So a lot of uptake, and I'm glad we have one of our solutions. And this is just getting larger and larger and larger. A lot of uptake on the timsTOF technology and what it can do for proteomics in general. So what we like to do is, back in the day when I was doing my proteomics at Scripps, my biggest pain was my sample prep was one company, my LC was another company, mass spec was one company, and my data analysis was another company. So if I didn't get the data that I needed, and I did not want to get embarrassed in a group meeting, I didn't know who to call, right? That ship has sailed, folks. Bruker provides a one-stop shop in terms of proteomic solutions. We partner with our friends at Proteomics And they're really excellent sample preparation kits for serum, tissue, etc. And their automation with the beadbox. We have our own line of HPLC columns with PepSep, columns directed for high sensitivity applications, high throughput applications, as well as single cell proteomics. And in the single cell proteomics realm, we are partnered with a company called Selenion, which have a single cell sorter. So the sample prep is standardized. It's taken care of, making sure there are very little batch effects. As you know, you have one of your our partner HPLCs, the EBSEP. We have our own HPLC, the nanovolute, as well as the mass spectrometer over here. But over the years, and with the genomics folk would appreciate that the more data you generate, the complexity of the data gets more, and it gets more and more difficult to extract biologically relevant information. So we have our own bioinformatics platform called Pazer. And I'll talk a little bit more about that later on. So if you have to do proteomics and you walk up to Griff and say, hey, I've never done proteomics, I want to start doing proteomics today, what do I need? We can provide the entire realm over here. You don't have to go to multiple places, and we stand behind the data that's being generated from everything over here. So that's what Bruker stands for. We stand behind the data, and we provide the complete solution in terms of these workflows. And when you listen to Kate's talk and Erica's talk, it's similar, that you don't have to go to multiple places. Your job is to answer these biologically relevant questions. And as a tools company, we give you everything that you need to actually get the conclusions that you want to draw. So what is TIMS? TIMS stands for Trapped Ion Mobility Spectroscopy. The initial technology was developed at Billerica, Massachusetts, not too far from here, in collaboration with Dr. Matthias Mann at the Max Planck Institute, with the first description of it in 2015, after which it was made into a product. So a lot of you are familiar with, or some of you are familiar with ion mobility in general, because it's been here forever. But trapped ion mobility, as you will see, is very fundamentally different, and it's better. And kind of addresses the problems with ion mobility in the sense that of cycle time, sensitivity, etc. So this is the innards of the system. And this is important to understand, because it plays into a lot of the issues that proteomics has faced over the years, such as robustness and speed and throughput, that this system actually addresses. So the ions are generated over here, and they're introduced into the system through a glass capillary. And this bit of engineering over here does a 90-degree turn. What happens over here is that this is a deflector plate. Anything that is uncharged goes into the vacuum. Only charged particles actually get inside the system. And this purple bit over here is the TIMS. This clever bit of engineering ensures that you are not introducing solvents or just your buffers and everything into the system, keeping the system incredibly clean. Traditionally, the problem with doing proteomics is that you're spraying things down the throat of the instrument, and as a result of that, system gets dirty, and there's significant downtime, and you cannot do high throughput work, and you have batch effects. This clever bit of engineering actually addresses that, making sure that we do a 90-degree turn on the ion optics, and only the ions of interest go in, keeping the system very, very clean. Beyond the TIMS, it's a standard QTOF. It's got an octopole here for focusing, and then eventually the quad, collision cell, and the TOF and the detector. But the real magic, folks, happens over here. I'm going to talk a little bit more about that and how TIMS is important and how the separation works and why it's groundbreaking technology. So imagine this. So this is the TIMS device, the purple bit that I showed you. And this is the entrance funnel, and that's the capillary making the 90-degree turn. So the way trapped ion mobility works is ions are introduced into the system, right? And they're pushed into the system through a steady flow of source gas, nitrogen in this case. As the ions get introduced into the system, their flow is retarded by an electrical gradient, a DC potential, right? Now, this electrical gradient makes sure that the ions prevent these ions from going right through. But these ions, because of the gas flow, they act like sails on a ship. So if you have a clipper, as you see over here, they have a lot of sails, and they can trap a lot more of the wind, and they can go further down the ocean compared to a small little boat like this. We're in Florida. We're all familiar with boats. So the ions act the same way. The larger ions trap more of the gas. They have more power to fight against this gradient, and they penetrate deeper into the TIMS device. Smaller ions do not have that much gas behind them, do not have that much energy, and they fall behind. And very neatly, they separate themselves as a function of what we call the collisional cross-section area. Fundamentally, it's their shape in gas phase. Larger ions deeper into the system, smaller ions fall behind, and they separate themselves as a function of collisional cross-section area because of this ion mobility characteristics. Now, we've separated them, great. What do we do? Now we elute them. And the way we elute them is by dropping this electrical gradient in a stepwise fashion. And you can see over here, these ions elute as a function of their collisional cross-section area. And not only do they do that, they also have a space and time concentration effect on them. That means ions together elute as discrete peaks. This boosts the sensitivity of the system about 30 times more than you would have in a beam type type instrument, right? This time and space focusing ensures these ions are eluting as a function of their ion mobility and then being pre-concentrated just like a chromatography column and making sure that we have a huge boost in sensitivity. Two things to remember here. One is have that clever bit of engineering, the ions degree turn keeps the system clean. And because we have this time and space focusing, which adds to sensitivity, we do not have to introduce gobs of material into the mass spectrometer, keeping it very, very clean, making sure that you can run day after day after day without any downtime. So this is how basically TIMS works. Now, the discerning among you will notice that while this elution happens, you cannot introduce any more ions on the front end. And that is a problem. As a traditional mass spectrometrist, I own all the ions. I want to use all the ions that are being generated. We do not want to throw any ions away because that destroys your duty cycle. So what does one do? One comes up with a clever solution where you put two TIMS in tandem. You have an accumulation device over here, which accumulates the ions, and you have an elution device over here, which elutes them as a function of ion mobility, right? This is where passive comes in. That's parallel accumulation and serial fragmentation. And this is kind of how it works. So your ions are being introduced again, makes a 90-degree turn, and they're accumulated for 100 milliseconds in the accumulation TIMS. That's a lot of ions, okay. There's a 2-millisecond transfer to the elution device. And now these ions are eluted as a function of their ion mobility. And while these ions are being eluted, in parallel, a second set of ions are being accumulated in the accumulation device. Now, as these ions are being eluted, this is what we call the standard MS1 survey scan. And the quad actually records the order of elution, what ions come out when. Now, when the second packet of ions actually come in, they're transferred. And now the quad steps through and does the fragmentation because it knows exactly what's coming out. But while this fragmentation is happening, another set of ions are being accumulated in parallel. That's where parallel accumulation and serial fragmentation comes in. And this happens over and over and over again, making sure that 100% of these ions that are being generated are being used. That improves the duty cycle because we're not throwing any ions away. And this is important because when you have precious samples, everything that you prepare laboriously, introduce into the mass spectrometer, all of that is being used. The TIMS technology and the passive technology over here, the workflow, makes proteomics much easier. And I'll tell you why later on. So traditionally, we've done a proteomics, which is pre-dedicated on three variables, as it were. Master charge ratio, obviously, MS, and retention time, right? These are the things which we relied on traditionally for the last 15 years to do bottom-up proteomics. Now, the problem is, any of us who have done any sort of chromatography, is that retention time is fickle, right? Retention time changes because of the age of the column, the quality of the solvent, the temperature of the column, or if it's a bad Saturday and if it's raining. It just does. Such is life. So relying on retention time is a bit problematic. But because we have ion mobility and because we can generate accurate collisional cross-section values, we can use the fourth dimension, as we call it, to add more to increase confidence in our quantitation and qualification. So this addresses two fundamental problems with doing proteomics the traditional way. One, as you imagine, this is the problem with all sort of mass spectrometers. If you have an ion, if you have a species, a peptide or a metabolite or whatever, which is similar in retention time and mass with another peptide, it is nearly impossible to separate them without long chromatography and fancy chromatography. This has been the bane of mass spectrometry for as long as it has existed. Ion mobility comes and actually resolves that to a certain extent, because you might have species that are identical in mass and retention time, but depending on the post-translational modifications, it behaves very, very differently in the ion mobility sphere. And number two, we don't have to always rely on retention time. We can use collisional cross-section area as a fourth dimension to add confidence. That makes our lives a lot easier. And I'll give you some examples about that. So consider these two species, right? Completely different peptides, but same mass, 825, and same retention time. Under normal circumstances, the mass spectrometer would consider that as one species. Because they behave so differently in gas phase, you can see over here, because of ion mobility separation, now the mass spectrometer knows that it's a different species, and this results in very, very different MS-MS spectra. Under normal circumstances, this would be a hybrid, what we call a chimeric spectrum between these two peptides. Because we are able to separate them in the gas phase, folks, we see really, really different spectra, and we identify them as different species. And this becomes important for post-translational modifications, because something like this, identical peptide, but differentially phosphorylated. Good luck trying to separate this on biochromatography, right? But think about this. The retention time over here for this guy is quite different. No problem separating this. But these two, on the other hand, elute at the same time. Identical mass, as you may imagine. However, they behave very differently in the ion mobility sphere, the way they act in the gas phase. And because of that, they're very well separated when you do an extracted ion mobilogram, right? And you can integrate under that peak, under that mobility, you can get very different spectra, and you can identify exactly what the site of these phosphorelations are based on the differential MS-MS spectra. You will be unable to do that on any other system, because this will not separate, or you need to be an amazing chromatographer and take a lot more time and effort or derivatization to actually separate this. That, folks, is the power of the TIMS technology for PTM characterization and overall IDs. And I think you're doing a little bit more of that in your own lab too with EvoSep. So traditionally, with mass spectrometers and proteomics, we've done DDA, and Bruker is doing really well in the DDA with what we call mobility offset master aligned, that's ion mobility separation. We're also able to do DIA, right? Data independent analysis, label-free quantitation, unbiased label-free quantitation, and obviously targeted analysis with PRM, parallel reaction monitoring. So we cover the range of workflows, making sure that whatever your flavor is, we have it, and we can give you the solutions for that. So let's talk a little bit about data-dependent analysis and the data that comes out of it. So this is our standard experiments that all of us run. These are HEK cell line digests, and you can see over about 200 nanograms on a 70-minute gradient, we get about 7,000 protein groups. Great, fantastic. 200 nanograms, 70-minute gradients has been the standard of QC for as long as I remember when I was at Scripps. But Bruker shines in running lower amounts because of the time and space focusing that I mentioned in TIMS, and running fast. So at 20 nanograms and at 30 minutes, we still get about 4,500 protein groups. What the system shines in is running fast gradients, low amounts, and getting good depth of coverage. Run low, run fast, run deep. And the competing instruments cannot do that because of just the technology, because they need more accumulation times and dwell times. They need to run longer gradients. You're running 2, 3, 4-hour gradients, generating huge amounts of data, which takes another 2, 3 hours to analyze. This, on the other hand, makes your life much easier. You're running low amounts of sample, keeping your system clean. No matter how sensitive the mass spectrometer is, how robust it is, if you shovel a whole bunch of material in a mass spectrometer, it is going to get dirty. There's no doubt about that. Because if you're running 20 nanograms, you'll be running fine. No problems. That is the key to running this system. And this is a classic experiment, right? Post-translational modification analysis by Julian Langer and Max Planck, right? He takes about 10 million hippocampal neurons, does the workup with enrichment titanium dioxide and IMAC, And then he splits these samples and runs it on the timsTOF Pro and some mass spectrometer that we will not talk about today. And this is an apples to apples comparison, right? Fundamental PTM characterization. We love phospho work. So let's have a look. And he analyzes this via three different search engines: Sequest, MaxQuant, and Peaks, right? Because search engines matter. And you can see, irrespective of the enrichment process, irrespective of the search engine, the timsTOF outshines the competing system, and that is because of the MoMA. That's because of the ion mobility, the ability to separate ions that are isobaric in nature, and also using collisional cross-section area to ensure that we have more confidence in our IDs. This is some work done by Professor Stefan Denzer. He's doing some great immunopeptidomics work if you're interested. But this one was done on a timsTOF Pro 2. And you can see that when you're doing phosphoproteomics, right, what we call epiproteomics, he was able to identify 27,000 phosphopeptides, of which 4,000 were isobaric, right? Positional isomers, like I showed earlier. And of them, about 2,000 were co-eluting. And he was able to separate 457 just via TIMS, no fancy chromatography. This, folks, is a bit dated. He can get up to 700 phosphosites now with his current technology. And this gets more and more and more better. Doing PTM characterization could be phosphorylation, could be glycosylation, acetylation, ubiquitination. The timsTOF really shines. This is what's in our wheelhouse. So let's talk a little bit about data-independent analysis. And why do we do DIA? Everyone used to do TMT. Because there are problems with TMT, because there are problems with DDA in general. DDA, its peak picking nature is stochastic, making sure that the peaks that you pick in February 2022, there is no guarantee that the same peaks will be selected by the mass spectrometer in 2023 February. As a result of that, when you do large population studies, like what Genomics does, large population studies, you cannot do if there's batch-to-batch differences like that. And this is the problem with tandem mass tags. Because of DDA, you cannot, the longitudinal studies get really, really challenging. But data-independent analysis is the same group of ions over and over again. And you're ensuring that you're utilizing these ions correctly and utilizing all of them irrespective of their intensity. Because DDA peak picking is also dependent on intensity. And we all know as biologists, the most interesting peaks and the most interesting species are not the most abundant ones. Nobody really cares about albumin in plasma. I really could give a toss about IgG in plasma. But I'm interested in those little biomarkers which are low level that actually tells me if somebody has leukemia or somebody has colon cancer. Those are the fun bits, right? And this is where it comes. This is where DIA comes in. And this is where plasma proteomics and high throughput proteomics is important. But we all know among us experts, DIA has been along forever, right? I mean, it's been with Sykes, Waters invented it originally, and then Sykes did their open swath. The problem with DIA is obviously not only ion utilization, but data analysis. And Bruker has actually solved that problem, because traditionally DIA is when we walk across multiple mass ranges and then analyze everything that is in that mass range. The problem with that is if you have, again, co-eluting isobaric species, the mass spec will be unable to isolate correctly and give you the right spectrum. So the way we do DIA is different. Not only do we separate based on mass, we also separate based on charge and collisional cross-section area. And this becomes important in standard bottom-up proteomics, because when you do bottom-up proteomics, your fun bits, the ions you're interested in are in the 2+ or 3+ range. You don't really care about the 1+, because that's chemical noise. Most cases, you don't have a choice. You have to focus on the 1+, and then that wastes your time. But because 1+ charged ions behave differently in gas phase than 2+ and 3+, you can actually separate them out, as you can see with the ion clouds. This is 1+, 2+, and then 3+. And you can only tell your mass spectrometer to focus on the 2+ and 3+ ions and use them for MSMS. And you can do discrete windows by drawing polygons around it, making sure that only the ions of interest are actually used for MSMS. You still collect an MS1 spectrum for everything, but you spend the time doing MSMS only on the ions of interest over here. Now, this is important, very important, because if you're doing other applications, it could be metabolomics, could be immunopeptidomics, where your charge states are different. For example, if you're doing HLA characterization, you have high masses and at 1+. These polygons can be moved anywhere in that entire spectra, and you can only focus on the regions of interest. You do not have to waste your time doing MSMS on species that you don't care about. This is really, really critical, and you're not filtering anything out. You're letting all the ions in, but you're only focusing on the regions of interest, making sure that the MSMS spectra that you get is rich and is actually relevant. No point fragmenting 1+ ions in this case. And what does it look like? This is what it looks like. At about 12 samples a day, this is done on the EvoSep actually, on 200 nanograms of HeLa, you get about 7,300 protein groups. You get about 9,000 now, but this is a bit old. But 60-minute gradients are good, but do you really want to run fast? So you want to do 300 samples a day, that's about 5 minutes. You get about 3,400 protein groups. Not a lot of loss for 5-minute gradient. So if you're doing high throughput plasma screening or protein degrader work, you want to run at about 5 minutes per sample, right? Again, run fast, run low, run deep. The system excels in running really, really fast gradients. Gone are the days when I used to do proteomics on the bench, running 3-hour gradients, starting the run, going for lunch, having a beer, and coming back, and the run still going. Do not drink beer for lunch. Sorry. So this is the key for in Bruker's proteomics solutions, right? And this works for DDA, this works for DIA, better ion utilization, making sure that we do not lose a lot of coverage, even if you're running fast. Data completeness is also important. You want to make sure that your batch effects are minimized, that the run after run after run, you're identifying similar or the same peptides and similar and the same protein groups. And this is critical, again, for population level statistics. And you cannot do that on a DDA-based run over multiple years. The reason genomics is years ahead of proteomics is because of the robustness and the throughput nature of its sequencing platform and the ability to handle that data and draw population level biological inferences on that. We are lucky that we're running 300 samples a day, but my dream is that in the next 3 to 4 years, we'll be running 3,000 samples a day, and we will be able to do a population proteomics for entire regions and understanding risk effects from cancer and leukemia and stuff like that. Another boondoggle for proteomics has been plasma. The problem with plasma, it's nasty, and it's got a huge dynamic range. There's a lot of albumin, there's a lot of IgG, but the real fun bits are obviously the low level. And we've had depletion strategies to get rid of the more abundant species. And the problem with that is that with albumin is for lack of a better word, is attached to a lot more other proteins and peptides. So when you actually crash out albumin, you take all of that out. And the batch-to-batch effects of crashing out albumin are terrible, right? Because you're not crashing out the same proteins along with albumin every time. Again, so statistics cannot be done on them. The CVs are bad. That the plasma has been the third rail of doing proteomics. Nobody wants to do it, but now we can. Because we work with another company called Seer Bio, it's based out of California. They have these lipid nanoparticles. Sorry, not lipid nanoparticles. They have the particles over here, which got antibodies in it, and then it enriches specifically certain species of proteins that you're looking for, getting a huge depth of coverage. Again, this is not depletion. This is enrichment. You're not crashing anything out. Everything that goes in plasma, it actually goes into the system. And at 90 minutes, we get about 1,700 protein groups. Traditionally, non-depleted plasma, you'll get about 500 if you're lucky. But at 90 minutes, we get about 1,700 protein groups. But again, at 8 minutes, we get about 600. An 8-minute gradient in plasma, 600 proteins is not too shabby. Again, run deep, run fast, and run low. This is a study done by Dr. Akhilesh Pandey. He's got about 3 or 4 systems at Mayo Clinic, and this was done in the heydays of COVID, if one can call that. He's taken some swabs from people's noses and throats, and he's done some DIA analysis. And this is what it looks like, right? He's done it with competing systems earlier, and you can see he's getting different kinds of proteins. But with the current study done on the timsTOF, he gets significantly more protein IDs. And the good thing about it is that there is a lot of overlap. So he's not looking at things which is completely out of left field, which shows that what he was looking at, he's still looking at these right now, but he's also looking at a lot more other protein groups that has not been identified earlier. And there's obviously he can do the statistics and draw biological inferences based on that. And again, it needs throughput, and it needs proper data ion utilization and ensuring that you're identifying the same things over and over again. And then you can actually talk about the differences between biological samples rather than worry about differences between analytical runs, because that's been the confounding feature of doing large-scale proteomics over the many years. And that brings us to our timsTOF HT, cleverly named HT because it stands for high throughput. One of the problems with doing timsTOF runs on the timsTOF nanosystems is that because the trap time mobility device is fundamentally an ion trap, you are limited by what you can load on it, right? And if you have a sample which is really heterogeneous in terms of their protein content, you might see all the more abundant stuff, but the less abundant stuff kind of falls through because you just can't detect it. So that's what our customers really complained about, that you cannot load a lot on the system, and that's why in 2022 we launched the timsTOF HT. So the HT obviously is an iteration of the Pro 2. So it's got the fundamental hardware difference is that it's got an XR TIMS cartridge or extended range, and it's got a better digitizer. So the original system had a 10-bit digitizer, and this one has a 14-bit digitizer, making it much more faster. So it can do about 150 MSMS, 140 to 150 MSMS per second, and it's got better quantitative accuracy. So what is the difference in terms of data? So if you're running a timsTOF Pro 2 or a Pro, you can load from about 3.12 nanograms to about 200 nanograms. You get about 5,600 protein groups. But if you start loading more beyond this, your protein IDs and peptide IDs are going to plateau, and then it's going to crash. So you can't load anything. There's no benefit to loading more than 200 nanograms on a Pro 2 and HT. However, on the Pro 2, sorry. On the HT, because of the extended range cartridge, you can go all the way up to a microgram, and you can see a corresponding increase in the amount of protein groups. And that plays well into DDA as well as DIA. You get about 10,000 protein identification on a standard cell line. A standard human cell has about 12,000 proteins, give or take. But with the DIA analysis and DDA, you can get about 10,000 proteins, again, on a 60-minute gradient. So this is the reason the proteomics world is really, really pushing on unbiased label-free quantitation via the data-independent analysis. And that plays to biological inferences too, right? Because this is some work done by Dr. Florian Mayer. He was a former pupil of Matthias Mann at Max Planck. Now he runs his own lab at the University of Jena. And he was able to analyze some cardiac tissue. And cardiac tissue, myocytes, et cetera, have a lot of heterogeneity in them, right? And you can see on his DIA analysis, he was able to load 200 nanograms and get about 3,500 protein groups. But when he loaded 800 nanograms, his number of IDs went up. And not only did the IDs go up, he had a dynamic range for about 5 orders. So not only is he seeing more, he's also seeing wider. He's seeing the more abundant stuff over here, and he's also seeing the less abundant stuff. Because we all know with disease states, with biology, the less abundant stuff, the less abundant peptides, the less abundant biomarkers are the ones that are really interesting. And this is where the timsTOF HT nanosystems actually play in. And finally, I'm going to talk about the timsTOF SCP. And I really do not like the word SCP, single cell proteomics system, over here, because it really does a disservice to the system itself. Among the high resolution mass spec instruments in the world right now, this is the most sensitive system around. Nothing comes close. Right? And that leads to applications where you're sample limited. Obviously, single cell proteomics, you are sample limited. Immunopeptidomics, you are sample limited. You don't have micrograms or nanograms of samples. You have picograms of sample, right? So if somebody takes a tumor biopsy, right? That's what, and so I'm based out of Boston. We have a lot of cancer centers, the Koch Cancer Center. So when they do tumor biopsies, that sample is a few nanograms, and that gets split between multiple departments, radiology, pathology, as well as the fundamental research. So the kind of actual sample that you finally get in your hand to do mass spec analysis is a few picograms. And you have to draw your conclusions from those few picograms. And that's where this system actually plays into really well. So what does it look like? If you remember my second slide, it's got the same the bit over here going on from the TIMS. What makes this special and different is that the ion capillary, so the glass capillary over here has a larger hole in front. As a result of that, more ions come in. As a result of that, more sensitivity, and then it does 2 90-degree turns to ensure that only the ions of interest go in. And to deal with more gas, more ions, there's a high pressure ion funnel and an ion transiting multipole to focus. Beyond this, the system is identical to the Pro 2. There's nothing, no difference. It's got the same robustness, it's got the same trap time, mobility separation, same collisional cross section. It's got a brighter source. We've introduced more ions, making it a far more sensitive system than what is available currently. And it leads to multiple applications. Obviously, single cell proteomics is one of them. Needle biopsies, like I mentioned, immunopeptidomics, and if you and this ties into what Kate's going to talk about, imaging. If you image a tissue, right? What we do currently, we take a cell, we take a tissue, smash everything up, blend it up, and put it in the mass spec. That's great. We can see the proteins and peptides of interest, but we lose the spatial information. We do not know where these peptides and proteins of interest lie in a tissue, and that's important. And Kate will talk about why that's important and how all of this builds into what we call spatialomics. But you need to have that sensitivity to analyze the regions of interest after you image it, and that's where the SCP system comes in. So again, single cell proteomics, one of the reasons we've been struggling with this for the longest time is obviously, mass spectrometers are not that sensitive, and it's a pain to analyze, to process the samples, because your starting material is so low that anytime it touches a pipette, you lose some. So if you think you're taking a single cell and you're injecting whatever you extract out of it, I don't know, 20, 200 picograms into the mass spectrometer, because of all the workup, you lost 90% of the material. But this is what Bruker tried to address, because again, if you want to do single cell proteomics, you walk up to Bruker, we have the complete solution over here. All you have to provide us is the cell suspension. And we worked with a company called, again, based out of Europe, called Cyanion, who have this, who has the system called the Cell-in-One, which actually sorts the cells for you into this proteochips, which then go on to our LC, which goes on to our mass spec, which is then analyzed from our bioinformatics platform called PASER. Again, data is good, but if you cannot extract biologically relevant information out of it, it's useless. It's a fancy toy, right? It's like having a Ferrari without wheels. It sits in your garage. So what does the data look like? So on a standard cell, human cell has about 200 nanograms of material. 200 picograms of material. But if you do an analysis or serial dilution of HeLa cell lysase, you can see that at about 250 picograms, we get about 2,500 protein groups. This is dated. Currently with the current technology, we get about 3,800 protein groups, right? But we can go all the way down to 62.5 picograms, about a fourth of that material, subcellular amounts, and we can still see 826. And it really scales well when we go up to about 32 to 50 nanograms. If you have micrograms of material, this system is probably not for you unless you're willing to dilute it, the samples. But if you're challenged with the amount of material that you can have, this system is perfect. And this brings to some of the groups that I talked to who are doing proteogenomics, right? They have a single cell. They want to do RNA-seq, but they want to do lipidomics, metabolomics from that cell, and they want to do proteomics, and you're limited in sample because you want to drop the proteogenomic information together, and this is where this system really, really shines. This is done on EvoSep that you obviously have in your lab. But 50 samples a day is not good enough. We have a method coming up with EvoSep at 80 samples a day. But look at that. We have not lost a lot of depth. And even at 200 picograms, we get about 803 protein groups. And at about subcellular levels, we get about 227 protein groups. Again, run fast, run deep. And this is where the Bruker systems really, really shine. And dynamic range. Maybe a lot of times you're not analyzing a single cell, because when you look for tumor heterogeneity, you can, or myocardiocytes, you actually work with 1 cell, 4 cells, 5 cells, 50 cells. And it actually has a dynamic range to handle that. And we also want to make sure that one single cell has about 250 picograms. You can reanalyze just the cell lysate and then one cell based on the sorting, and the results are almost identical. So that's where the system really, really shines in terms of dynamic range as well as sensitivity. And this is some data that just came out of our collaboration with Evosep. These cells were actually sorted based on their shape. So this is what the Cell-N1 can do. They can actually sort the cells based on what they look like in solution. And that gets important because remember the cardiac tissue experiment that we did? Myocardiocytes are not homogeneous. They are differently shaped, they have different functions, and the Cell-N1 can actually separate these myocardiocytes based on their shape and then do a quantification and qualification of the proteins. This is some, if you get a chance, this is some great work done by a researcher on the West Coast at Cedars-Sinai called Dr. Jenny Van Eyck. She's really pioneered this kind of workflow where she separates myocardiocytes from patients and then quantifies the proteins of interest and peptides, and is directed towards understanding why people have heart attacks. And this is critical. We can also do immunopeptidomics. This is some work done by Professor Steve Carr at the Broad Institute. Again, he was not doing single cell proteomics back then, but now he is, and he's analyzing some immunopeptides that were pulled on via antibody enrichment, and you can see based on current technology, the timsTOF identifies about 36% more peptides. This is one of the most challenging experiments in proteomics because these immunopeptides are rare. They're small, but they're indicative of infection, of cancer, of any sort of perturbation on the cell. And getting them out of the cell without destroying the natural state of the cell is really an art. And then this mass spectrometer actually plays in really well because we can actually use the material that's presented to the mass spectrometer and get the peptides and proteins of interest. The similar experiment, again, previous technology with the timsTOF and compared to the timsTOF, we get about 32% more peptides on the SCP. And this gets better and better and better as we improve the methods, as we improve the TIMS resolution, etc. Finally, my last bit is going to be about bioinformatics, because this is where genomics is streets ahead of us, is that taking large volumes of data, because now I've told you that I can really run really small amounts of samples and I can run it fast, so I can generate a ton of data. Well, the data is useless if I can't process it correctly, if I cannot draw biological inferences, because ultimately this is where we are at. If I can't go up to an MD and take my data and explain to him or her the relevance of that data to their work, none of this really matters. This is just a fancy toy. Again, Ferrari without wheels. And this is where Bruker's proprietary search engine comes in. We call it PASER, and this is the last acronym I'm going to throw at you, I promise you. It's called Parallel Search Engine in Real Time. And what does it really do? One of the problems with doing mass spectrometry-based proteomics is that it takes forever to analyze the data. But in this case, the PASER is basically a standalone computer that sits right next to the acquisition computer. And while the acquisition is actually happening, the data is transferred real time while it's happening into this box. And as soon as the run is done, you get the results out. So you get the protein groups, the peptide groups, you get the MS spectra, the MS spectra, and the collisional cross-section values. You get the PTEMs, you get all sorts of information that you would normally get from MaxQuant or Mascot, but all done within a minute of the run getting done. So now you don't have to wait days to get the data that you need. You can also do a couple of other things. All of us has been through this, right? We leave on a Friday evening, we set up our QC, we set up our blank, and we set a whole bunch of runs up. We come back on a Sunday night, or hopefully not, or a Monday morning, and we realize that the QC was complete garbage. All your samples are useless, that the runs that you've done. And you have to do prep everything again. What the system can do is you can put parameters on it saying that your QC on a, say, 90-minute gradient, you want to see 5,600 protein groups. If it doesn't see that, because it's doing it in real time, if it doesn't see that, it'll stop the run right there because QC not met. If your blank is dirty, it'll realize that right away, it'll stop the run and it'll not proceed, making sure that you're not wasting your samples, you're not wasting your time generating a bunch of data that is completely irrelevant. This is the power of PASER again. And this comes, and almost everyone who's bought a timsTOF Pro 2 or an HDLR systems have gotten PASER and they use it as a QC tool. It can also do other things. So we've used data sets, and this is from some work done with Professor Stanley Stevens at USF, Griff? Yep, University of South Florida. We've taken huge data sets of post-phosphorelations generated on Stan's system, and we have trained the AI, the artificial intelligence in our box, to be able to recognize and generate collisional cross-section values based on peptides with particular phosphorelations or acetylations. Now, what it does is we generate a collisional cross-section value from the system, which is measured, and we compare it to the predicted collisional cross-section values, and we use a matching algorithm to make sure that the right peptide is actually identified. This is a problem because normally what we call FDR calculation, false discovery rate calculations, we have a list of 5 most probable peptides which matches a particular spectra, and we pick the top 2. If that spectra MSMS is bad, then you have a false identification. But the collisional cross-section value, which is actually calculated and measures, is intrinsic. And because we can compare these two, we can generate what we call a TIM score, just like an A score, to ensure that we have more confidence in the identifications we're doing on these peptides. This is important again for PTM characterization. We've done it really well with phosphopeptides with STAN. We're training the system on ubiquitination now, acetylation, and eventually glycosylation, to ensure that the IDs that you're getting, you're far more confident in it than you would do with just MS or MSMS. And as you can see, Stan is absolutely thrilled over here because he's been able to get about 30 to 35% more phosphopeptides identified just based on the collisional cross-section value and TIM score. Again, this is done by DDA as well as DIA, and you can see with TIM score and match between runs, you get significantly more IDs confidently identified. If you didn't have TIM score, if you didn't have collisional cross-section values, you will not be able to do that. And finally, something that's really, really critical here is that Bruker's data format is open, right? That means we have a lot of third-party vendors who can actually use our raw data, our native files, our collisional cross-section area, and generate data results. Some of them paidware, like Peaks, but others are free, like MS Frager, for academic institutions at least, and Diane, Vadim Demichev's work. Also, if you have your own a bioinformatics pipeline, we will give you the data file. We'll also give you an SDK, what we call a software developers kit. And you can rip it apart and you can run it through your own pipeline the way you want. This is what the Broad Institute did with Spectrometer, that's Clauser, right? So the idea is that we have nothing to hide. Our data format is completely open. You can use it in any way you want. You can use any sort of software, and it'll give you excellent results. With that, I will leave you with this gentleman over here. His name is Raphael Helig, and he's really, really thrilled because he's just got done running about 3,000 nasty plasma samples. And during this time, he has never had to clean the system. This is in Roman Fisher's lab at Oxford. Now, this is the norm. We have another gentleman at Boston Children's Hospital, Professor Hanno Steen. He's done 8,000 plasmas, nasty COVID plasma samples, without the system going down. This is a testament to the robustness of the system. Again, we run low amounts, we run fast, and we do deep proteomics on the system. With that, I will give it up to my colleague, Erica, and we'll address all the questions at the end of the talk. And thank you, everyone, once again for your time. It's a pleasure being in Florida.